I guess while we're getting set up there, I just want to introduce myself, Mike Kelly. I'm President and CEO of NervGen. I've been with the company for just a little bit over a year. I wanted to thank Brian, the Lineage team, the Reef Foundation, all the sponsors of this program for inviting me to speak today. Also wanted to thank a few of our clinical advisors that are in the audience. I'm going to be talking about a clinical trial today that they helped design. So appreciate their input. We are also a public company, so this is my lawyer's favorite slide. I just wanted to say that I will be making some forward-looking statements, and all of the risk factors for our business can be found on cedar.com. So who is NervGen? Here are the highlights of our company. We're a small company. We're virtual. We're based in Vancouver. I'm based in Philadelphia. My chief medical officer is based in LA. My head of discovery is based in Boston. So we are a child of COVID. We've actually learned how to work independently, and we're on Zooms pretty much 5 to 10 hours a day. We have two key programs within the company. NVG-291, which is our lead product candidate that has moved out of preclinical into clinical, is what I'll talk about today. And we also just announced earlier this week on Monday, NVG-300, which is an internally discovered molecule that is actually moving into preclinical work with in ischemic stroke, ALS, as well as spinal cord injury. But NVG-291, I think Dan Michael last year presented at this meeting, and he showed a lot of our preclinical data. This product is a trial that came out of Jerry Silver's lab, and I'll talk about the history of the technology in a second, but it has been demonstrated to show that it can repair the nervous system, it can restore motor and sensory function, and it also can restore cognitive function, all in animal models. Now we have to see if we can translate this into the human. So the big news of the day is we are in our 1b/2a clinical trial today, and that's really the main focus of this topic. This is the team that is virtual and all over the planet. We all have 30-plus years experience in the pharmaceutical industry in various different functions and capacities, in developing products and commercializing products and discovering products and getting products into clinical trials through the FDA and approved. My recent experience before joining this organization, I was part of a team that developed and launched Narcan nasal spray in the US and Canada. So we actually helped save many lives, and it was really challenging. After that company, we actually sold the company to a bigger company who took the product to the next level and saved many more lives. So when I was searching around for my next opportunity, I had a pretty high bar personally that I wanted to achieve, and NervGen really checked all the boxes for me. A little bit of history and some of the reasons why I joined the organization, why I think a lot of people invest in this company. We're a pretty unique company. We're public, but we're public in Toronto. We're retail held. So not a lot of biotech companies are held by retail investors. Typically they go down the path of a venture fund, an institutional investor, and then they'll dip into the NASDAQ pool of biotech institutional funds. I would say right now about 70% of our shareholders are people like you and I, everyday investors. We're held by retail folks. We have an extremely strong base of supporters that have been sticking with us, and it's really a not traditional path for a pharmaceutical company to go down this path. So. And one of the reasons why I think we have such strong support is because of the technology. So Jerry Silver is the founder of the technology. It came out of his lab. So Jerry's in the bottom left-hand corner is a picture. If you ever met or talked to Jerry, he's crazy, he's passionate, and he is a mad scientist. And if this works, he will get a Nobel Prize for this technology. I'm thoroughly convinced. So what Jerry discovered back in the 90s, when he was focusing, actually, before I joined, I had a long discussion with him, and he started out looking at the optic nerve, and he was trying to wonder, well, why does the optic nerve go in a straight line? Why doesn't it grow all over the place like every other nerve or nerve system? And what causes nerves to grow in certain places and not grow in other places? So I said, Jerry, he was using all very big terms. I said, Jerry, you got to really dumb this down. I'm a business guy, and I'm learning the science, and so he did. He explained it very simply to me. He says, there's a group of molecules called CSPGs, chondroitin sulfate proteoglycans, right? And they inhibit repair. Those are the guardrails. Those are what force nerves to grow in a certain pattern or not grow in other areas. Those are what cement nerves together in your spinal cord. Those are what hold things together in good times. But when you have damage due to injury or disease, CSPGs inhibit repair, right? So they protect, but they also inhibit repair when there's damage. So that was the first big discovery, that CSPGs are very, they're in abundance and upregulated in the glial scar, right? So the next big discovery was through Jerry's collaboration with Harvard. And they tried to figure out, well, how are CSPGs talking to cells? And they discovered PTP sigma, which is the receptor where CSPGs will attach to cells and send the signals in to do this inhibition. So then Jerry's team, one of which, Mark DePaul, who's now in our company, he was the leader of the team, who started to look at different types of products that could actually interfere with this signaling. And they discovered NVG-291. NVG-291 doesn't specifically attach to the receptor. It actually goes into the cell, and it disrupts this downstream signals. So in animal models, the Nature paper has been published to show that it actually can help regenerate, right? So that was the first big publication. So how did NervGen come to be? So Harold Punnett, who's our founder, he's a dentist, scientist training by background, had a misfortunate event occur in his family. His daughter-in-law took a fall, and she was paralyzed. So Harold took it upon himself to scour the earth for technologies in development, try to find meetings like this, try to look for different companies who had different things. Came across Jerry's Nature paper, started a relationship with Jerry and talked to Jerry for quite a bit of time, flew out and had many meals with him, and then convinced him and Case Western to in-license the product or out-license the technology to him and a group of investors in Vancouver. And that was the beginning of the company. They since then went public 1 year later, hence we're down the retail path, and then now we are in human clinical trials. So that's the background of the technology. I mentioned we have two different programs. When I joined a year ago, we were moving 10 balls down the court, 3 steps at a time. And that's typical of a small startup company. But then once you pick a lane, which is the top green lane, which is spinal cord injury for us, that's where the real money starts to get spent. So the purple bars are $150,000 to $200,000 bars. The top green bar is a $9 million bar. So when you pick an indication, that's when you start to spend money. So my job is to educate retail investors why we're spending so much money. We're spending so much money because we're actually trying to prove that this product actually translates into humans. So those are the indications we're targeting. We had to put Alzheimer's and multiple sclerosis on the back burner for now, but we will certainly circle back once we get proof of concept in this trial. So Dan Michael spoke here last year, and I stole a few of his slides because not all of you were here last year, but several of you were. But I wanted to talk a little bit about what Dan and our clinical advisors did to help design this clinical trial. So the first thing that Dan taught me is, well, Dan came from Amgen. He's an expert in designing clinical trials. He's an expert in drug development. And he said, well, you don't try to prove something that you haven't seen proven before. So everything on the left-hand side of the page is known and has been proven preclinically. So everything on the right side of the page is how we're approaching this clinically. So we have data in the acute model, we have data in the chronic model in the rodent. So we're studying chronic and we're studying subacute. The animal model studied motor incomplete, that's why we're studying motor incomplete, and so forth and so on down the page. The next slide shows you a little bit about the design of the trial. So the trial is a commitment. You know, for people living with spinal cord injury and the talks that we heard this morning, I'm almost embarrassed to say how many tests we're putting folks through and the commitment that we're asking them to do. But we're more than halfway there with the first cohort of patients, the chronic cohort patients. And this is a major commitment. They're moving to Chicago. We're a single center study. It's 16 weeks long, 12 weeks of treatment, 4 weeks of observation post-treatment. And why we're asking them to stay there that 4 extra weeks is because in the animal models, we still saw improvement after the treatment period ended. And that's a big known. And if we could prove that, that's one step closer to proving to the FDA that we have a drug here. So it is single center. We partnered with Shirley Ryan Ability Labs. I have to give a shout out for Wings for Life. I know they're not here, but they were very instrumental in helping us get this off the ground. They've actually are covering a third of the cost of this trial, which is a major step forward for us as a company, but for this program in general. We couldn't have gotten it off the ground without them. The main reason we chose Shirley Ryan, there's 2 reasons three reasons we chose Shirley Ryan. Number one, Monica Perez. Monica Perez is an expert in electrophysiology, and Shirley Ryan is one of the best rehab hospitals in the country. And there is a rehabilitation component to this, and electrophysiology is our primary endpoint. All right, so I mentioned there's 2 cohorts, 20 patients in each. The first cohort is chronic, defined as 1 to 10 years post-injury. The second is subacute, and we just are changing that definition now. We're actually amending the protocol. We were submitting an amendment to the IRB next week to push the window from, originally it was 7 to 49 days, and now it's 20 to 90 days. So it broadened it a little bit. But it also, I think, gives us a chance to get folks who are being discharged from their first set of rehab an opportunity to come to Shirley Ryan and still meet the criteria. Before, it would have been challenging for somebody to be discharged from a hospital and get to Shirley Ryan. So some of the inclusion criteria and exclusion criteria are on the page here. I think probably the biggest thing that was presented in one of the earlier presentations was the funnel, right? How many people do you have to put in at the top of the funnel to get somebody who's qualified at the bottom? That was the biggest learning for us in this trial. And it's still an ongoing learning as we move into finishing out the chronic and moving towards the subacute cohort. But the key entry criteria, I think that is probably the biggest challenge is we used to have a screening process. Now we have a pre-screening, and then we have a pre-screening, pre-screening, screening, and formal screening. So we've broken up screening because it doesn't make sense to have people fly to Chicago and spend 4 days for screening and have them miss 1 or 2 criteria. So we do most of our screening virtually, and then the final screening that they have to be in Chicago for is electrophysiology. So everybody entering into the trial has to have signals. The primary endpoint of the study is change in signaling from the brain past the injury to a hand muscle group and a leg muscle group. So there has to be a registered signal in that hand muscle group and leg muscle group to enter into the trial because we're measuring the change in that signal as the primary endpoint. So that's the objective, the primary objective. We're measuring 10 different muscle groups in general, but those 2 are the primary objectives. And it's a small study because Monica and her biostatistician and Dan had sat down to figure out mathematically, well, how many people do we actually need to see a change in electrophysiology? It was actually 8 per arm. We bumped it up to 10 because we also have a ton of secondary endpoints here. And these are all the usual suspects of secondary endpoints and quality of life outcomes. And thank you to, I thanked Jared earlier. We added another twist, very similar to the AbbVie group after last year's meeting. We took the feedback. And so we partnered with a group called EMIS. It's actually, the original company was Casimir. They were purchased by EMIS. I think the owners of Casimir may be buying the company back, not sure. But they specialize in listening to people with lived experience And they specialize in, the cartoon says it all. Not everything that counts can be counted, not everything that can be counted counts, right? So when we're doing an exploratory study like this, we're looking to find a signal. So we're looking at historical things that have been done, and we're trying to innovate and look at new things that have come out. But the thing that was missing was the gaps. What's between the gaps, which is lived experience feedback. So we've actually are submitting this as a protocol amendment as well, not only to help design the questionnaire, but to hopefully quantify that questionnaire. And this group has experience. They've worked with companies that have actually gotten drugs approved through the FDA using this methodology. So this was presented at Asia about a couple weeks back by Dan. This is the first 12 subjects. Obviously, this was submitted in April. So we're past this, but it does give us some confidence that we're on the right track. Obviously, you could see that the average age is typical to the average age we've been hearing from the presentations today. It is skewed heavily towards male population versus female. But you could see that, everyone does have a baseline MEP amplitude, which was critical for this study. No dropouts so far. So far, everything is going well, knock on wood, and we're still casting the net and trying to get the word out nationwide to get folks in the top of the hopper so that they can come out the bottom and live in Chicago before the winter comes. So in summary, we're advancing NVG 300, as I mentioned earlier. NVG 300 is a new molecule. We're not talking a ton about it because as a small company, until you get all of your IP protections in place, you really don't tell the world what it has, so you beat around the bush a lot, and you kind of tell them why you're happy about it without telling them really what it is. We will start preclinical trials in the next month in 3 new indications, and hopefully in the fourth quarter of this year, we will be announcing the results of those early trials. I will mention the SCI trial is a confirmatory trial. We've already done one study in spinal cord injury with NVG 300, so the next study is a confirmatory trial. That will tell us whether we have a drug candidate or not. So we are a 2-product candidate company now, rather than a 1-product candidate company that we were last year, sitting here. We're well funded for the next 2 years. We're well on our way to hopefully getting the chronic enrollment complete in Q3, which will have a data readout in Q1 of next year. And then sub-Q will start to enroll as we end enrollment in the chronic cohort. Thank you very much.